So let's look at the causes of and limits of resolution in bold fMRI. An advantage of fMRI, as I said, is it's large scale. We can measure the entire brain every 1 to 2 seconds, which is great for extracting information that's distributed across systems. But how fine-grained can it be? And what information are we getting by improving the resolution or losing it by not doing so? So spatial resolution can in principle be relatively fine-grained. It's limited by, in part, the bold point spread function and the signal to noise, which depends on a number of factors. Among these are the field strength of the imaging, so higher field is better on average, the distance of the head coil from the tissue being assessed, the underlying vasculature, and how fine-grained the distribution of neural responses are draining into particular blood vessels. The upper limit is probably somewhere around 400 microns. So this would be high field, high resolution imaging, sometimes with implanted coils like Nucos logothetis and others have done. So by implanted, I mean implanted under the skull on the surface of the brain. Can't do that with humans. So an example using human fMRI is visual cortical imaging at 7 Tesla. And this study is interesting in 2 ways. So they're looking at the visual cortex, which we saw an example of earlier. We know that it's retinotopically mapped. And this means that what Polemani et al. Were able to do is figure out what do we have to show a person so that the mapping under the cortical surface is give us the shape that we'd like. So they have this distorted looking letter M, and they're presenting alternating checkerboards of where these M's and then the field around the M's are alternating. And what that should look like on the cortical surface is the letter M. And when they do the imaging, they can actually see there the letter M on the cortical surface, as expected. So that's one illustration of how fine-grained the resolution can be. And what's also interesting about this study is that they're interested in signal from different lamina, different layers of cortex, which all span about 1.5 millimeters or so, give or take, across the thickness of the cortex. So here you're seeing signal from the deep layers ranging to the superficial layers. So it is possible in some cases to image specific layers, which is a very exciting frontier. So let's look at the bold point spread function a little more closely. It's related to the microvasculature and to the bed area that's affected by local neuronal activities, and it varies with the field strength. So at 1.5 T, the bold point spread function is relatively coarse. It's about on the order of 4 millimeters, 3 point something to 4 millimeters. At 3 T, it's on the order of 3 millimeters, and at 7 T, it's on the order of 2 millimeters or possibly less. And measuring those point spread functions is an ongoing process. Another question, though, is what's the effective resolution in a group analysis? Most of the time, we care about a group of subjects, and everyone's brains are different. So the typical effective spatial resolution for a standard group study is much lower. It's 10 to 15 millimeters, sometimes more. And why is this the case? Well, for one thing, artifacts cause image distortion. Secondly, we're lining up all the subjects' brains together, usually, and those subjects' brains are very different from one another. They don't all line up anatomically, and there are errors. There are also individual differences in functional anatomy. So I'll appeal to Truett Allison's work here on electrocortical mapping in humans of the fusiform face area, and he shows very compellingly that it's variable across individuals. And then finally, there are diffuse modulatory effects. So often our bold signals aren't influenced by local neuronal activity, but potentially by very broad patterns of neuromodulators that can affect bold signal in many areas of the brain at once, causing very blurry blobs. Some additional issues are a concern the spatial alignment between subjects. So there are many sources, and within subjects as well. So there are many sources of mismatch. We have to line up, as we'll see in the pre-processing lectures, the functional images and the structural images. And that mapping is not exact. In particular, the functional, often EPI, echoplanar images, are distorted, so they won't match what the anatomy looks like even for that person exactly. Another issue is spatial misalignment between the structural and the template or reference image. So we often align images to an atlas space, and that alignment is not perfect. And the atlases themselves are also approximate. So the famous Taylor-Ack and Tourneau atlas of 1988 is a very widely used atlas even today. And this atlas was based on one hemisphere from one woman who was a 60-something-year-old French woman. And they took one hemisphere and they did some set of architecture studies. And then they assumed that the other hemisphere kind of looked like that one. So when you talk about Brodmann's areas that came from that set of architecture in the Taylor-Ack atlas, those Brodmann areas are very imprecise. So we can't be sure about where they are. So what does this mean for us? Well, there's a few take-homes. One is, if you're doing a study, create a mean anatomical image from your study and use that as an underlay to display your functional results over, because that's going to be more precise than using an atlas. It's going to be a better picture of where your activations really are. Doing that is also an easy way to see some problems with intersubject alignment. If all of your brains look different, there'll be some ghosting in that image. Finally, the take-home is, especially in group studies, interpret the anatomical locations of your results with caution. There are some new solutions. I'll talk about two things here briefly, and they'll come up in later modules when we talk in more detail about these things. One is this idea of hyperacuity. And this is the idea that the pattern of activity across the voxels might contain more information than any one voxel by itself. And that means if I'm using multivariate pattern analyses, I can sometimes detect functional topography, like ocular dominance columns, for example, even if the voxels are not small enough to fit within any one functional area. So voxels will span multiple functional areas, but that's okay in some cases. And the second idea is the idea pioneered by Jim Haxby of hyperalignment, which is to avoid the idea of averaging anatomical brains together and assuming that everyone's brains have to fit in the same space, and instead have everybody watch a movie and then use the movie data across the brain to put people in a common functional space. And he's shown very interesting results that show that it can produce much better cross-subject matching and classification results than anatomical alignment. So that's a new frontier as well. I'll talk a little bit more about hyperacuity. So in the brain, neurons are often not randomly intermixed. As I said, there's functional organization at multiple spatial scales. However, even if those neurons are randomly intermixed, as they are in many types of cell assemblies, in the visual cortex, in the auditory cortex, in the amygdala, there can still be information that you can extract from fMRI about those different population level responses of intermixed neurons. So this is a little simulation that I did with 3 kinds of randomly distributed intermixed neurons in a spatial area. And so on the left, you see 2 event types that are intermixed with a homogeneous Poisson process. So they're really randomly intermixed. In the middle, you see another kind of random process, which is a Poisson random field, where essentially the hit rate determined by the Poisson distribution varies across the field, across space. And on the right, as the Poisson random field becomes more sparse or higher variance, you end up with more patches of clumpy neurons by chance. So what does that mean? That means that random distribution doesn't mean uniform, the same across space or across brain space. So the differences in neural activity at the fMRI voxel pattern level can be detected for all 3 types. And Amir Shmuel has done some really excellent modeling work on this, and his papers are referenced here. So here's a little simulation. As I said, the bold response most closely tracks the local field potential, LFP. So let's consider our 2 types of randomly intermixed events. These are the most mixed. And here they are. Random, in this case, though, is clumpy. So if we look at the distribution of simulated local field potentials, which are smooth, essentially representations of the neurons, we see different patterns for event type 1 and event type 2. And if we look at those patterns here are uncorrelated. So even if the neurons are randomly intermixed, the the local field potential across space isn't going to be uniformly distributed. And if you sample that at the voxel level, that leads to voxel patterns for the two types of events that are actually still, in this case, largely uncorrelated and are on average for this type of mixing. So that means that we can still classify these two events even if the neurons are randomly intermixed. I'll finish up this lecture with a little bit about temporal resolution. So EEG and MEG and optical imaging methods have much greater temporal resolution at the millisecond level. fMRI resolution is limited by the slow hemodynamic response function. This means that the onset of a response can be detectable often within 2 or 3 seconds, and the peak of the response happens at about 5 or 6 seconds. And we've seen this before with these nice, beautiful curves from Martin Lindquist's earlier study. This doesn't mean that all is lost. So there is this idea of temporal hyperacuity, and one way to do this is by averaging events. So if you average events of the same type, of type 1 and type 2, then you can detect a difference in the average response latency, even of 100 to 200 milliseconds. And this was actually the case in Martin's study, where he showed that these two curves have reliable differences in latency for visual and motor responses, even when they occur 100 milliseconds apart, either the visual before the motor or the motor before the visual. And one of the things that helped make this possible is trial averaging combined with rapid sampling methods. So he developed a technique here to collect whole brain images in 100 to 200 milliseconds. And this is now becoming a big part of the field with simultaneous multi-slice or multi-band imaging. So this is becoming increasingly popular and possible. So if we go back to this idea about the spatial and temporal resolution of human neuroimaging, we can see how we're starting to be able to push the boundaries. So with a standard 1.5T or 3T fMRI and group analysis, we can detect differences or information in relatively coarse maps and large-scale networks, on the order of multiple centimeters, most likely. Sometimes on the order of a couple centimeters. The best case at 3T is single subject decoding, multivariate pattern analysis, talk about that later, with high spatial or temporal resolution, and averaging across trials to improve the detection power for each of those. We can't do both very well. We can either be fast or we can be high resolution, but it's hard to do both. And if we really push the boundaries, the best case fMRI so far in the field is high field 7T imaging with single subject decoding and rapid sampling, and we can potentially do better in space and time. And why is this important? Because these are the levels of spatial resolution at which we really start to see lots of information processes and brain representations mapped. So there's a real hope that if we can push this boundary, we can really see more of where the real information action is in the brain. So that's the end of this module, and see you next time.